Thanks, Chris, for the lovely introduction. Welcome, everybody. It's quite sobering, actually, to be here celebrating something that's 40 years old, and I feel a lot older than it. So, my involvement with the Brain Bank is since 2002, so I've been around for 22 years. I don't know where that time has gone, although my daughter was home from university this weekend, so I know it's tied up in all of that as well. So life has a tendency of going back very quickly and passing by. So I thought I'd start by giving you a little bit of an overview into some of the history as I've learned it over the years, and then give some examples of the sort of research and the impact that's come from this that I hopefully will be of interest to some of you who are quite new to your relationship, and a trip down memory lane for some of the others. So this was where it all started. Gordon Wilcock came to Bristol in the mid-80s. And the story goes, although the Human Tissue Authority by today's standards would be very nervous to think about it, was the Brain Bank started in Gordon's boot of his car when he came from Oxford to Bristol. He'd already started his career in Oxford in memory clinic work there and was following up his patients and asking them for brain tissue, because this was in the early stages of development of the early cholinesterase drugs, which are used today. The galantamine was the first one being developed, which I think Gordon was involved in some of the early trials, and drugs like donepezil, rivastigmine, and so on, which are used in commonplace clinics today. Gordon's some of the early work of brain tissue was involved in some of that. I think the other thing to bear in mind too is that when he was coming, he kind of saw not only the value of trying to understand the disease more, which we really didn't know a lot about. The one thing to bear in mind about the drugs that are available at the moment, and I'll come back to the ones that are just going through the process of being licensed recently, is they were only treating the symptoms. Unfortunately, the diseases were continuing in the brain, and they had limited capability of keeping the function maintained in these people, because the degeneration of the brain tissue was continuing underneath, and those drugs didn't really do anything to stop that. So it was really only trying to top up some of the deficits were caused. Then the next person who was integral to these kind of strong foundations is Seth, who's hiding at the back as usual. He's very kind of understated way. So Seth, I think, came, was it about 7 or 8 years after Gordon, or around a little bit? I was 87, okay. So I know Gordon had some interactions with some of the neuropathologists, but when he latched onto Seth and Seth kind of met up, there was a meeting of minds, and the two people had a very, very strong ethos towards the benefits of translation. And translational research, which you've often heard about, or may have heard about, from us anyway, and at least in Gordon's and Seth's eyes, was all about trying to get something started in the tissue, and hopefully it would have a benefit in later life to patients. So the real bench to bedside adage. And one of the key things that Seth brought as a neuropathologist was started to introduce really rigorous, standardized ways of assessing brain tissue. But also, Seth was very research active, so he was very conscious of what the developments were in the field and tried to adopt those into the Brain Bank, which is basically the foundations, the procedures that we still use today and in which we're actually famous for. So that Gordon and Seth were obviously the kind of key linchpins in terms of the strategy and setting things in place. And then Sonia Matthews, who has been, she's still in the brace fold. I met her recently. She hasn't changed much either. She's making me feel particularly bad that I've aged a lot worse than she has. But Sonia was the Laura of the original Brain Bank. And this little humble building here was at the back of the day hospital at Frenchie, when Frenchie Hospital was still a hospital. So it was a bit of a 3 up, 3 down type of establishment. And the labs for the Brain Bank were just kind of tucked in the opposite corner there. And needless to say, we grew out of that pretty quickly in terms of the Brain Bank needs, but also the Dementia Research Group was based there as well, which was a small group. And then Laura came. And who you're going to hear more about, so I'm going to try not to steal too much of Laura's thunder. She's got her own story to tell. But I remember Laura coming for an informal chat, sitting in Gordon's office when we had advertised for what was Sonia's replacement. She was looking to retire at the time, and Laura was just finishing up at the University of Bristol, having done her degree there. And then obviously the rest is history, and you'll hear more about that later. In 2006 was quite a landscape, like a landslide shift for us all, because the Human Tissue Act of 2004 came into play. And the backstory behind this was, some of you may be around or may have heard about the Bristol heart scandal, where there was the retention of children's heart tissue in the central hospital in town, and it kicked off this national exercise and new legislation to ensure that consent was very much at the heart of tissue retention for research, which hadn't been particularly adopted very well, and which what triggered the scandal. So this was really quite a massive shift in terms of how tissue banking was going to be conducted in the UK since then. And what had happened was, for quite a long time, the Southwest Dementia Brain Bank, as a research resource, and also in terms of the Dementia Research Group, they were kind of hand in hand. We shared the same building, as you saw. There's a lot of crossover between the two. But after that legislation came through and the Human Tissue Authority was set up, we were forced through some of the requirements of that to kind of separate the brain bank a little bit more into its own kind of self-governing identity, and then into the research group, which we now call the Cerebrovascular and Dementia Research Group. It used to be the Dementia Research Group, but that's just to reflect the kind of direction of travel of some of the activities we've now moved into in the last decade or so, but the name change is very recent. But fundamentally, even though we're kind of operating organizationally and in terms of different levels of governance, there's still that very intimate interaction between the two. And that brings us up to 2014, where we moved to Southmead. So as you saw, we had a pretty humble building which was built, and it was the original building was called the John James Building, funded by another Bristol charity, the John James Foundation. And now we moved into the first floor and the second floor and of this end of Learning and Research Building. And the team obviously has grown, with Laura kind of being the start of the new group coming on. And some of the people here in the audience today, they can't, so that you can name them, shame them, and welcome them. But I particularly like this one, where it was kind of like the raised eyebrow look. I think she wasn't happy me asking for the photo in the first place. But fundamentally, the one thing that, and actually just to flag, 2014, I just realized when I was putting the slide together, it's 10 years since we've been in Southmead as well, as of May this year. So it's another little mini anniversary within that. But fundamentally, everything that's happened from that point, even to 2014 and beyond, has been only possible due to Brace. And obviously this is the old logo, so this is a trip down memory lane for the old charity members as well. And the elements are similar. You've got a suspension bridge, but this was deemed, if I recall the discussions at the time, to look more kind of molecular and related to the biology that the charity was funding. So that the investment just in the brain bank itself and provision of the resource to make tissue available for research, it also invested in researchers, as it's doing still to this day. And this is just some of the people it's invested in the Dementia Research Group of old and the Cerebrovascular and Dementia Research Group of now. And one of the earliest people it's invested in is Katie Chalmers. I think Katie was one of the first PhD students of Gordon's and Seth's. Is that right, Seth? And Katie is still in the university working in population health sciences. Scott was one of the first postdocs we hired. I think it was one of my first Brace grants back in 2003, 2004. And he's still with us. He's the co-lead of the group as it stands now. And he's got his own fantastic area of research, which he's growing with his own PhD students and research fellows. There's Laura, who we've mentioned a few times, and we'll be hearing more on. Jen Palmer. Jen was another Brace PhD student, and she's also working at the university as a lecturer. She's just back from maternity leave fairly recently. Taya, who's here in the audience, she was another PhD student. Rachel Barker, another PhD student funded by Brace, who's now currently working at Southmead. And Rachel's project is an interesting one because she took a career break to have her family, came back, started working with the cancer research group in Southmead, and now is doing a project funded by the Prostate Cancer UK as a research fellow, looking at the crossover between cancer and mechanisms and Alzheimer's disease. So she's retained her PhD origins in Alzheimer's disease and combined them with her newfound area of research in the cancer space. Ellie O'Sullivan, who was working in the brain bank until recently, has just started a PhD. And then, of course, Liz Coulthard as well, who is, she's now the chair of dementia neurology. She came to Bristol, I think, in 2014, 2015 as well, and set up the memory clinic, which is adjoining Brace. I've just realized I have a quote I have to read out. Have you got that to hand as well? Okay, great. Thank you. So at the end of the day, fundamentally, as I said, partnership and collaboration to innovate has kind of been central to both the interaction between the brain bank and the research group. And part of that innovation and the linkage between the brain bank and the research group is as much about the dementia research agenda in its widest sense, but also to continue to improve brain banking. So one of the areas that Seth was kind of particularly involved in, and I'm embarrassed to say that when I started in the group in 2004, I really didn't know a lot about cerebral amyloid angiopathy, and I'd already been working in the field for 10 years. So cerebral amyloid angiopathy is, you've often heard about plaques and tangles in Alzheimer's disease. Well, amyloid depositing in the blood vessels is what cerebral amyloid angiopathy is. And Seth had been working on this for a number of years and was pretty much to the forefront of a bunch of investigators across the world developing ways of assessing it and trying to understand its relationship with Alzheimer's disease and around the pathology in which it causes as well. And cutting a long story short, those interactions which Seth had built up over years, we developed a new protocol for how that would be assessed in post-mortem tissue to standardize it across both the UK, and it involved a lot of people in the UK initially, but then more widely with some of the leading people internationally. And so this is a protocol that's used in a lot of brain banks around the world now. Why didn't that move on? Sorry. And I'm telling you about this because weirdly it's particularly relevant even now today. So taking it up to the modern day, and I mentioned earlier about the new drugs being approved. So aducanumab and lecanemab, which are two drugs that were developed to reduce amyloid pathology, to be disease modifying and serve as an improvement for the kind of symptomatic treatments that we currently use to date. The problem was they didn't get approvals in most places around the world because they had a lot of high side effects. And one line of inquiry and theory around this is the fact that it exacerbates or it interacts with people who have high levels of cerebral amyloid angiopathy in their brain. And essentially the side effects that people were getting were abnormalities in their imaging where they were getting evidence of bleeds. And one of the theories is essentially, in a layman's terms, is that the drugs are trying to help the brain clear out all this amyloid damage, and it's getting stuck in the blood vessels, and those blood vessels then are struggling, and the bleeds occur as a result. Donanemab, which is the third one that's come through, this was most recently released and has been licensed in the UK for more limited usage, which is a fantastic step forward for people with Alzheimer's disease. The only slight problem is, and I say slight very kind of sarcastically, is the fact that it's only going to be given to people who have one or no copies of the apolipoprotein E4 gene. And for those of you who are kind of familiar with this space, ApoE is the strongest non-causative genetic risk factor that causes Alzheimer's disease. So a high proportion of people with Alzheimer's disease have one or more or two copies of ApoE4. Generally one, because having two copies is reasonably rare. So this means as many as only 30 to 40% of all Alzheimer's patients at the moment are eligible for this new drug. So we have a ton of work to do to try and find out, can we still find a treatment that we can give to the rest of the patients? So the amyloid angiopathy story, even though it's been around for years, is now kind of very much to the center of what's going on there. And there's probably likely to be more research going on. So those consensus criteria that we've developed will probably play out in some of the studies that are going to be done in the future, which will be probably post-mortem studies of people who've had some of these drugs to see what the extent of damage has been and so on. Another example of the impact of research done in the brain bank itself has been about vascular effects on pathology in Alzheimer's disease, which are often overlooked and have been largely ignored conveniently. And it's partly because most of the historical studies of Alzheimer's to date, particularly when they recruit for clinical trials, they tended to have very select criteria which people would be enrolled into those trials, and they would exclude people who would have high levels of either pre-existing cardiovascular risk or history, because essentially they were trying to make the participants in the trial as closely representative of what might be a pure form of Alzheimer's disease. The reality, of course, is there isn't really a pure version of Alzheimer's disease, except for the very rare, unfortunate cases where people are carrying gene mutations that affect multiple members of families over multiple generations. So that's the nearest thing you'll probably get to a pure form of Alzheimer's disease, and they tend to be often earlier onset, although not always the case. But if you get those mutations, they definitely cause the disease. So essentially, in the vascular space, we started looking at this, partly because the topic I'll come on to next in terms of some of the research we were doing, where we were starting to identify biochemical pathways interested, or that we deemed to be relevant to some of the vascular effects we're seeing in Alzheimer's disease. But when we started to look at how vascular dementia was being studied at the time, and that's the old name now, vascular cognitive impairment is now the modern name for it, we found it was actually a bit of a mess. When we looked at the literature, there was 15 to 16 different ways in which vascular cognitive impairment was defined around the world, which isn't conducive to doing standardized types of studies or trials in the future, or even large-scale studies looking where you might want to look at the genetics and so on. So everybody needs to be working essentially to the same kind of protocols. So we embarked on a study to try and at least address the classification issues. And thankfully, the community and the vascular dementia community was very receptive to it. It's much smaller than the Alzheimer's community. And I think they had finally come to a point of frustration as well. And the interesting thing is that of many of these kind of named authors on this particular paper, most of them had their name against at least one of those criteria. So we kind of did the political thing. We made sure that everybody who had a vested interest was involved in that study. And then we basically wrote to everybody that had ever been involved in any of these studies. And we created what was called a VIX group. So we wrote to, I think it was nearly 1,000 people worldwide who had been researchers published in this space. Inevitably, you got email bounce backs, they'd moved away and so on. But once we eventually published this study and came up with a new consensus theory, which was done through a kind of an iterative questionnaire-based thing, people sent responses, and then we kept working back and forth with all of this community. And this VIX group, I think in the end, the total author list of this was about 350 authors from all over the world. It's the largest study of its kind. The weird thing was that was only a study to figure out how we're going to call it and how we identify the subtypes. The next thing was we need to try and come up with a way that we recommended people get it assessed in a clinic so they could be aligned with those classifications. And we did that as well. And this was a smaller version of the same thing, still 150 or so people, and that got finished. But what came out of that was a very active community, and we realized that we had the same problems not only clinically, but in terms of how it was assessed in terms of post-mortem. So again, Seth and worked with Olivia Scrobot, who was instrumental to the delivery of these two studies. And Seth went and got some funding from another charity, not Brace on this occasion. We can't fund everything, but it was still massively important that we had the brain bank support, because a lot of the brain bank tissue was involved in this study. And the VSING criteria was a new set of criteria, again, done predominantly with the UK neuropathologists develop a new protocol by which vascular cognitive impairment can be assessed at post-mortem. And why is that all important? Because that's kind of now laid the foundations for this. So the naming of some of these gets a bit funny. So genome-wide association studies are where you get these big consortia and they get as many DNA samples together from people who have a disease type and a control. And off the back of our kind of vascular cognitive, we developed our own consortium looking at trying to get DNA together where we could do a genome-wide association to try and find new genetic risk factors. There's another consortium in the US called CHARGE, which was doing something similar. And we were kind of doing them in parallel with the idea that we'd kind of look at we try and replicate each other's findings. Long story short, we didn't find much, but it was kind of, there were some issues with some of the analysis which we left at the time, but we've come back together now with the rationale. We've developed a new methodology in which to approach this. The fundamental thing we have done, which was different to before, was we looked at the whole cohort in its entirety just to see if anything was coming out. And even though that was a little bit underwhelming, when we started to look at people, whether they had the risk factor gene for Alzheimer's, ApoE4, we found actually 2 different profiles in 2 different patient communities again. So almost a similar story to what we're seeing with Alzheimer's disease in relation to these drugs. But obviously the combination of the 2 studies is absolutely massive. And this is going to be the only study of its kind done realistically, because we will have exhausted pretty much every available DNA sample there is, apart from Emma's project, which is looking at this in relation to post-mortem. It will be a subset probably of these. But this is involving a study of 10,000 patients with 860,000 controls. There's data on that many controls from all around the world. Now, 10,000 patients doesn't sound a lot when you compare it to Alzheimer's disease studies. And that's true, but vascular cognitive impairment is much less common than Alzheimer's disease. And when I was telling you about how people tended to focus on Alzheimer's in preference to vascular cognitive impairment, this is kind of demonstrating a consequence of this. But this is ongoing. This is currently kind of a live project. And last week I was giving a brief update on this, and we have found so far 2 genes that are coming out of this. And this is an interim analysis because we've got cohorts from Iceland, Denmark, and a few other US cohorts to still go into the final analysis. But ApoE comes out in this cohort as well, and BIN1, which is another gene that has come out in the Alzheimer's genome-wide association studies as well. And what's kind of reassuring is this one that's come out now, called 4A1, which is very non-descriptive as well. It turns out that's a gene that's come up in stroke and cerebrovascular disease risk. And that's kind of reassuring because you would expect people with vascular dementia to potentially come with some stroke background or some disease that's related from a vascular background. So in some respects, it's almost like a positive control, and it's telling us the study is working. We hope to finish this study in the first half of next year and publish it. But as I said, it'll be the only one probably of its kind for quite some time. And then brings me on to just the last bit of the thing, which is I just want to give an example of a kind of a case study just to show how Seth and Gordon's plan for translation can actually achieve quite a significant amount of progress. So I'm going to harp on about this, which apologies to some of the team who are here. They've heard a version of this already in the last couple of weeks, and others who've been around longer have heard many versions of this over many years. So if I do see some nodding off, I know who they are, but I won't feel particularly upset about it. So the renin-angiotensin system, I'm going to introduce to some of you. It's a mediator of both the vascular and the non-vascular facets of Alzheimer's disease. So just to kind of give you a quick summary of Alzheimer's disease that a lot of people may already be familiar with, but historically, the main kind of things that are known is the plaques that we see above, the neurofibrillary tangles, and we see the very pronounced differences between an Alzheimer's brain in terms of atrophy and bleeds, and a healthier brain section of the same age. This here is a representation of the cerebral amyloid angiopathy, which I mentioned. So you can see that really black kind of circular mark. That's the amyloid embedding itself around a blood vessel. So that's a cross-section around a blood vessel. And you can see the damage that it's doing all around the environment of the blood vessel. And then all these other characteristics of things we know happen in the process of Alzheimer's disease. And it's partly the reason why Alzheimer's disease is such a difficult disease to treat. It's not just amyloid, it's not just tangles or CA. There's all these other effects going on. So It's almost like a house with many fires on in many different rooms. And it's probably no surprise that over the last 30 years, you have numerous different hypotheses have been generated to try and explain what is going on with Alzheimer's disease. I don't think it's too contentious to say that amyloid is probably involved in a lot of these, and that's why these new drugs are looking to target that, because the rationale is if you get this, you can address many of these as well. And I don't think that's particularly contentious, but it just shows the complexity. In terms of the vascular aspects of dementia, so this is just to highlight the fact that A, it's overlooked quite a lot, and yet of all those characteristics I've just described, those that are circled in red are areas where vascular dysfunction has a major impact. And even some of the major hypotheses that have been proposed have a very strong vascular component. And that was the neuropathology. When you just look at the epidemiological kind of profiles of these, and there's numerous different versions of this that have happened over the years, depending on whatever studies you've done. And this is your typical pie chart of the proportions of different types of dementia. And as you can see, vascular damage turns up in lots of these slices of the pie, causing its own problems. And in the clinic, this is very, very common. And quite often people who've both an Alzheimer's disease and a vascular dementia, they have a more aggressive form of the disease. They tend to decline much more quickly as well. So some of the work then we were doing in the brain bank focuses on your kind of classical profile of what are deemed modifiable risk factors, something we can try and do something about. And it relates to hypertension, which is high blood pressure. When you look at the vascular element again, though, there's a vascular component to numerous ones of these as well. So it's there to stay, it's unavoidable, and it's something we need to understand more about. And then the other just little nuggets just to highlight is the fact that this is a kind of demonstration of the hypothetical time course of development of Alzheimer's disease from various earliest stages of pathology right through to somebody developing quite advanced disease. And the one thing that is a recurring observation is that it can be about 20 years or so between those earliest changes, or at least it's accepted that it can be up to 20 years between the earliest changes and then you see some manifest symptoms. And the age range at which that could start then could push it anywhere from middle age onwards. And middle age is itself even being redefined at the moment. It used to be 40, now it's almost 50 to 60 because of life expectancy increasing. But that sits right in the period where hypertension is also most common in the population. And then finally, this is a graph that's been proposed, and there's numerous versions of this knocking around, which is trying to demonstrate the cycle by which Alzheimer's maybe evolves in people. So first of all, when people are normal and younger, they start to develop some amyloid pathology. That amyloid pathology changes the environment of the brain such that tau will start to develop. And then these will combine with other factors to start to affect the cellular structure and the brain structure, and then the cognition and the kind of memory changes we all are more familiar with start to outwardly manifest. Again, in terms of the recognition or the lack of recognition of vascular changes, one reanalysis of the data set that gave rise to that previous slide looked at the vascular contribution to those changes over time. And it's a slightly different, very busy structure, but the key thing to just take home from this is that whereas the amyloid, the tau, and everything else I described is still represented in this, the first thing that changes here is a vascular dysfunction. So the theory there is that, again, it's probably earlier than even the amyloid. So just to take you through one of the pathways we've been looking at then for the best part of, since Scott Miner started actually, Scott and was started working on this with us 20 years or so ago. And it relates to this pathway called the renin-angiotensin system. Now, it's a bit of a busy pathway again, so I'll hopefully make this easy. So essentially what we have here is a pathway with a protein at the start, some enzymes that chop away at it, make smaller molecules, and then this particular molecule here, angiotensin 2, binds to one of two little receptors which does stuff biochemically. And one of the combinations here with this receptor will increase your blood pressure and this one will reduce your blood pressure. So we were interested in that to see if this was a mechanism that might be related to some of the vascular changes we had in Alzheimer's. And partly the reason we were looking at this was before I came to Bristol, I'd already previously collaborated with Gordon when I was doing my own PhD, looking at this particular enzyme and gene variations in it that found an association with Alzheimer's disease. And the variants I had found that were increased in Alzheimer's appeared to change levels of the enzyme. So we thought, okay, we've got a genetic predisposition to the function of this enzyme, whether it has a bearing on this, that might have a bearing on this blood pressure regulation. The other thing just to put in mind with this, that this molecule here, which is the kind of focal point, it's also the centerpiece for another part of the pathway that looks to break it down and move traffic to other receptors over this way. So this receptor here, MAS receptor here, this is an alternative binding site for a product of this. And that has another benefit of also reducing blood pressure. So it's a really complex pathway because there's 4 receptors here, 3 of which have different effects on blood pressure regulation. And as I said to you, they cross over around this molecule angiotensin II. And when we started to look at this in brain tissue, we found that both of those sides of the pathway were vastly changed in Alzheimer's disease. So I'm going to give you a quick introduction to some of the things we had found. And on the left-hand side here are some of the studies we did on brain tissue from the Southwest Dementia Brain Bank. And we were the first to report this, where we found that ACE, the enzyme that makes that molecule I was highlighting, was hugely increased in Alzheimer's disease. And we found it in various different contexts and also supportive evidence. Unfortunately, there was also other studies going on elsewhere in the world showing changes to the same pathway that were also having effects that were also relevant to Alzheimer's disease. So we felt we were on the right track. And even in animal studies, they found that if you started to give drugs that interfered with that pathway, which already existed because they're used to treat blood pressure at the moment, it also had an effect on some of the pathology or the cognitive function that existed in animal models. So why would this be happening? So that's the thing when you're doing Translate, you have to keep going back and double checking whether things fits with the kind of array of information that's available. So when we look at our risk factors as well, we were trying to say, okay, if this angiotensin II and ACE pathway is highly regulated, is there evidence in other risk factors that exist that we can kind of pin to the same kind of effects? And long story short, as the slide is quite indicative of, this Ang2 molecule, it crops up in most of these other risk factors. So if that pathway is heavily changed in Alzheimer's disease, if it's also increased in some of these pathways that are risk factors, it makes sense that it's probably related to Alzheimer's disease, so that you have a kind of a cause and effect type dynamic going on there. When you look in the brain then to see what's the effect of the changes to those pathways in other systems, and this is a slide trying to crystallize probably several hundred different papers looking at these different areas in their own right. And you can demonstrate there's a lot of evidence supporting that Ang2 is involved in a very negative way in the production of a lot of these things, at least in a contributory manner. So the rationale is that we were proposing that because of that upregulation of pathway, Ang2 does feed quite heavily into a lot of these pathways involved in Alzheimer's disease. So we were particularly emboldened by all this level of data, plus a lot of epidemiological data that was existing at the time, and this is summary, some snapshots of evidence that was coming in epidemiological studies. And then way back, I think it was in about 2010, 2011, David Cameron, I'm not sure whatever your political persuasions are, but he did something really good as he got Alzheimer's disease on the agenda, and he insisted there was a lot of funding invested in doing trials. So we took that opportunity to try and do a clinical trial that explored some of the stuff we had found in the brain tissue was supported by all these other these other types of studies like epidemiological findings, animal studies, and so on, to see if we could really test it. And essentially, clinical trials are really, really hardly to find experiment. And that gave birth to the RADAR trial, which started in 2012 and only finished in 2019, thankfully just before COVID hit, or we would have been embroiled in that and an awful lot of trials which suffered from it. This was a study that involved 23 centres across the UK. We led it from Bristol. And it was a study looking at a blood pressure drug that targeted angiotensin II called losartan. Our primary outcome in that study, which was very ambitious, was we were hoping that the drug might have a protective effect and slow the rate at which the brain shrunk over 12 months, which is measurable by volumetric MRI. It's quite a predictable pattern once you get to a certain stage. And we also then had other kind of outcome measures which were secondary. The study wasn't big enough to be able to do them meaningfully. It would give us some indicative findings. The slightly depressing thing, even though I said it was a 7-year trial, is we found no evidence to support this, unfortunately, from the trial. And the rationale, the conclusions were that the patient group we had recruited to the trial were mild to moderate Alzheimer's patients, and they were just too severe. There was too much damage accumulated for these drugs to be able to offset what had already been set in play probably 10 years previously. There was one slight concern in the trial, because everything was so negative, and some people were trying to put a positive spin. It was consistently negative, which is meant to be reassuring. But we were worried, what happened if the drug wasn't working properly? And then we would have had basically a trial that was duff. But fortunately, in the people who had received the active blood pressure lowering drug, their blood pressure had reduced in the study of the trial. So we knew the drug was actually working. And that, I think, rescued the whole study, because otherwise, if we couldn't have demonstrated that, that study would have never seen the light of day in terms of publication, because we would have been given a duff drug. The slightly frustrating thing, having worked in the lab with loads of people on this over the best part of a decade, is this chap here who's bounced around from Boston to Emory and so on, and who I've met a couple of times. Ihab has been doing small-scale trials, and he has done now 2 trials of people with mild cognitive impairment, and he's found suggestion, and the drug he's used, candesartan, is very similar to the one we used. And even at the time we were designing the RADAR trial, it was losartan because it was off patent, and candesartan was just about to come off patent. And one of my co-investigators said to me, should we not use candesartan? I said, no. Let's stick with losartan because it's already off patent. We'll be waiting 6 months. And if we start doing this, the company might sniff it and they might want to try and interfere with it. So we said, we'll stuck with losartan. I wish I'd listened, because this guy has got suggestive evidence that it works in milder people. So that's really a bitter pill to swallow. But if anything, it's demonstrated how starting sooner in patients is better. So while the trial was rumbling on, we went back into the lab, of course, because there was very little evidence of what was happening in this other part of the pathway that's supposed to be protective in Alzheimer's disease. So we started to look at that as well. Technically, what we were looking for is this pathway should be impaired in some way, because then that would explain why the other side of the pathway was heavily increased. So when Scott and some students of ours started to work on that, we found what we were hoping for, actually. We found that people with Alzheimer's disease in their brain tissue, their activity of that ACE2 mediated pathway, which is the key enzyme in that pathway, was operating at half the level of controls. It was also reduced in a level that's, the more reduced it was, the more severe the pathology. And it was heavily correlated with the amyloid and tau pathologies. I'll skip this one, it's the same slide. So when we put the two together, the red indicates bad and the green indicates good, ordinarily for its function in Alzheimer's disease, with Ang2 in the middle. And what we have found in Alzheimer's disease, that the red pathway dominates and the green one is suppressed, and it basically does a lot of damage. That's the kind of working hypothesis. So with that hypothesis in mind, we started a different line of inquiry. And this is a study where we thought, well, if we could activate that ACE2 enzyme, could that have a protective effect? And this is the kind of spoiler alert, the heading gives you the answer. But essentially, we did this study with colleagues in Cardiff, where we found this compound, diminazine aceturate, DISI for short. You can see why it's easier to pronounce. So dizzy is a drug that's it was discovered in 1944. It was only used in the veterinary world. It's not licensed for human use, but it had a property of being able to enhance the activity of ACE2. So we thought, okay, if we can boost that green side of the pathway I showed you, it might have a protective effect. And so this was just a behavioral experiment where we found that we had mice that were given no drug, and their behavior was pretty much the same before or after a treatment period which we had developed. And similarly, then we had a mouse model, which they had in Cardiff at the time, that basically had an impaired function at the point in time. We were going to start treatment, and then they tested them again after the treatment period, and they found if you gave them no drug, there was no change, unsurprisingly. In the mice with Alzheimer's kind of pathologies, once we gave them the drug, their behavior recovered and pretty much was back the same to wild-type mice. Now, wild-type mice don't have any engineering. They're basically just normal mice. So we were kind of blown away by this. This was kind of a bigger effect than we were anticipating. When we looked at the amyloid pathologies that ordinarily exist in these mice, we found as well that particularly in the versions of amyloid beta, which we know cause pathology, the drug had a lowering effect on those, i.e. protective. And reassuringly, it showed as well that the drug was increasing the ACE2 activity, which it's supposed to do anyway, compared to the other mice where the drug wasn't administered. So this was really encouraging as a potential therapy. The final thing which, when you're doing in the drug discovery space, is you have to show specificity and mechanism. So we had identified there was another drug that can essentially interfere with that interaction we had been testing between dizzy and ACE2. It's a compound called C16. And long story short, when we basically gave mice with dizzy and this C16, we found none of those protective effects we had already found. So it cancelled them all out entirely. And because that C16 was very specific to ACE2 as well, we knew that what we were seeing, all those protective benefits, wasn't through some secondary process. ACE2 was integral to what was being changed. And we also found actually that in some of that, when we did a little bit more tissue work, we found that actually the changes to ACE2 were happening specifically in the hippocampus as well, which was also really important because the hippocampus, which is right in the center of the brain and one of the first areas to go in memory, it was also being upregulated. So this is a drug being given peripherally and having a central effect. We did two other sets of experiments. So the first experiment was just a 30-day treatment experiment. We did another experiment that was 10 days long, another one was 10 weeks long. And long story short, we had very similar protective effects. So it posed us the question of whether, that should be ACE2, sorry. Enhancement of ACE2 activity is a possible other treatment opportunity in Alzheimer's disease. And again, I just wanted to reiterate, we never would have looked at all of this prior to, if we hadn't had access to brain tissue. Now, this story is kind of slightly paused, because I don't know if any of you are up to date with the COVID pandemic. ACE2 might have sounded familiar. So ACE2 was the receptor for the SARS-CoV-2 virus that we all had our lives kind of altered with and caused COVID-19 for a lot of people. And that paper which I showed you, which we did with the Cardiff group, we published that in 2019, and we had applications into the Medical Research Council in 2019, and then a reiterated version. We submitted, I think it was December 2019, and the pandemic hit March 2020. And we honestly think, because we had ACE2 in the title for Alzheimer's, it just got chucked out because everybody was COVID, ACE2, and we didn't mention anything about COVID because we didn't know about it at the time. It landed after we submitted our grant. So we're waiting for the COVID dust to settle still before we can revisit this. But we feel we're on the brink of coming up with a potential new treatment option. And we've got some work going on with some colleagues in town using AI to try and identify already licensed drugs that may interact with ACE2 that we could repurpose in the same way we did with the radar trial. And it'll be a watch this space. So I'm going to finish up I have been asked to play for time here to some extent because Liz couldn't, and I'll get off in a second. So just the final reflections from being around for 22 years with the group as well. So Brace's support for the Brain Bank and the existence of the Brain Bank, at least locally, we've come up with some new areas of research of biological mechanisms that have eventually moved into clinical trials. That's the translational pathway in its own right. And that wouldn't have happened if we didn't have the Brain Bank. And that's just locally. Laura will be telling you later the amount of requests we get for tissue all over the world in the UK, where you're going to have a similar story potentially with new treatments. I think the Brace support we've had since the start, that's provided the infrastructure to also help us provide the new underpinning studies just for brain banking type standards generally in the UK and internationally as well. So that's kind of probably less well known because it's quite a niche activity area, but it's fundamental to making sure that the highest standards are adopted. The Brain Bank in itself, through its existence, has created massive opportunities for people to go and not only do new types of research, but also for people to build their own careers on. Some people will hear, and there's several people in the room here working the Brain Bank, and they're developing their careers off the back of that as well. And you saw some other people who I had, who've been through the ranks and moved on to other roles elsewhere. And just to say that, you know, like it's 40 years, this is a celebration of the Brain Bank, but it's also one of Brace. This is a legacy they share, and one I hope that will continue. As Chris said, I'm not sure I'll be around in 40 more years to do this sort of thing, but that's hopefully the foundations that we built up now. So, and the foundations are very strong. So thank you very much.